they are tolerating. Then they were about 130 LDL in the beginning. After medication of 2 years, it comes to around 43, 42 like that. And they are stable.
Okay.
But generally they feel there's a little weakness, and when they go for walk, a little cramps, all vague symptoms. Nothing
quite
really.
certain that's related to statins. Yes, they certainly do that. I think it's a question of balancing the risks versus the benefits. So for most people with primary prevention, I never push their LDL cholesterol to 40.
Correct. Yeah,
So,
it's only secondary prevention.
Yeah, so if the risk is between a second heart attack versus a little bit of cramps here and there, I
back again. The mistake is current didn't go now. It is Airtel. Kindly you get a better connection. Okay, fine. We'll move. We live in India. Okay, God bless. I must send the link to everybody. They have to connect the new link. A clinical for Sanjeevani ella to kalsprapa. Tell them join the new link.
Okay. So this was the thing. Okay, so this is it came out like about a year ago. Okay, probably less than a year ago on. So this is the outcome study of the PCSK9 inhibitor Evolocumab. Okay, so this created a lot of press and attention. So this is the FOURIER study. So basically, they took about 27,000 patients with established heart disease. So this was a secondary prevention trial. So 27,000 patients, 90% of them had had a heart attack, another 10% had had a stroke. All of them were on high intensity statin therapy. So their baseline LDL cholesterol was about 90. And they all got this Evolocumab. So there are two PCSK9 inhibitors. So one is Atherothrombosis and the other is Evolocumab. So this can either be given once every 2 weeks or once every 4 weeks. So they pooled both of those. So you can give them once in 4 weeks also. And so as you can see, so there was clearly a nice LDL lowering. So this is the LDL cholesterol level 30. Okay, so you can drop the mean LDL cholesterol down to 30 and versus this 90. So this is when the PCSK9 inhibitor is added on to statins, then you get this 90 to 30. And what was the result of that? So you can see this primary endpoint. So this is at the end of 2 years. So the mean period follow-up was just 2.5 years. This is what created a lot of furore and controversy. Most statin trials, most cardiovascular trials are at least 5 to 6 years. So here they found that the event rate was quite high, and they already had the adequate number of events at the end of 2.5 years. And so I think they just did it because it was, the problem is, most of these trials are really not done for science. They are just done for regulatory approval. Okay, so they just want the drug to be approved, and it's very expensive. So if you actually complete your trial in 2.5 years, what you're supposed to do in 5 years, so you're cutting down your budget by 50%. That's millions and millions of dollars. And that's probably the reason why they did it. So they stopped the trial short. So you can see the average duration is just around this. And of course, they stopped it because they already saw the separation. And so there is about a 15% relative risk reduction. Yeah. So the hazards ratio is 0.85. The absolute risk reduction is just about 1.5 to 2%. Okay. And to many of us, so that kind of was disappointing. Okay, because we really felt that if you can lower somebody's LDL cholesterol down to 30, you can expect it. Oh my God, we're going to see this would be the game changer. Okay. This is not really so astounding. Okay, this is just about what we saw in most other trials also. And is that because nobody knows? Is that because you stopped the drug study earlier? And then if you read this paper, okay, so more than a page of the discussion is on this. Okay, then they try to extrapolate and say, oh, if we had continued for 5 years, we would have seen this much, this much, this much. Should have done it. Okay, but they didn't do it. All right, so we don't know. And more importantly, I think the reason why they should have done it is safety. Well, we really want to know how safe it is to keep people at this cholesterol level for years together. And we don't have that data, unfortunately. But anyway, this is where we stand. And so clearly, yes. So does it reduce outcomes? Yes, it does. Okay. And some people say, oh, it's simply because these are all secondary prevention. So they already have established disease. So you can't expect like an 80% relative risk reduction or something. So no matter what, yeah, so this has now been approved, and so we do have this. But the reason why I brought this up is, so where do we draw the line? Okay, what should be the LDL target? And so there's one other smaller study which probably is more instructive, and this is a study done in the Cleveland Clinic. Okay, it was called the Glegor trial or something. So basically, it's the same thing. So people have established coronary artery disease. There's about 1,000 patients. So they got this monthly Evolocumab, and again, so what you see is that LDL cholesterol is 90 versus 30. What they did here is, so Steve Nissen at Cleveland Clinic, everybody there gets an intravascular ultrasound, and then they measure the plaque volume. Okay, so they looked at the regression of the plaque. Okay, so this is the on-treatment LDL cholesterol level, and so at different LDL cholesterol level, what was the change in percent volume of the atheroma? So you can see that once, of course, you get to this, if you have an even when you're on treatment, okay, look at this. So this is the beautiful thing. So if you have an LDL cholesterol above 90, the plaque volume actually keeps going up, all right, even when you're on treatment. So it really is, so just putting somebody on a drug and thinking, oh, I'm done, okay, is probably not right at all. So you will really see plaque regression only here, okay, when it's actually below 90. And to me, what was the most scary thing is, look at this. This doesn't seem to, it doesn't level off. Okay? So it's going down. So when you're at 40, so clearly, we thought, oh, 70 is good. Why is 70 good? So if you're actually at 50, that's much better than 70. And if you're at 40, that's much better than 50, and so on, so on, so on. Okay? So at 20, so this thing is still going down. This, I think, is the problem that we'll have to deal with. Okay? So when do we say, okay, that's it. If we lower it further, we won't see any more benefit. Okay? Because this is direct evidence of plaque regression. Okay? So we will see plaque regression going on as LDL cholesterol keeps going down. So obviously we can't get everybody down to 20 or 30. So it's a question of balancing the risks versus the benefits. I guess these are the questions that the expert committee is pondering, and they'll have to come out with some guidelines on whether or not to switch to LDL targets. If so, what should the target be? I think so. That's one of the points I wanted to make. So this is, those of you who have been to the US probably recognize this. So there's a whole chain of stores called the Target stores, and that's their logo. Okay, so we'll, as I told you, this stock is repackaged from something I'd given this thing. So we're talking more about targets. Okay, so what is an ideal target? Okay, to get, right? So one is, I mean, all along I've been just talking about LDL cholesterol, okay, as if that is the ideal target. Maybe it is, but then consider this case scenario, okay? So we have two patients, all right? So one of them has, this is his lipid profile. So 198, 130, 90, 50. The other is 210, 130, 250, 30. So basically, both of them have the same LDL cholesterol level, 130, 130. Does it mean that both of them have the same cardiovascular risk? Not really, okay? And why is that? Because we know that cholesterol is not just simply floating in the blood, okay? Instead, it is packaged in these lipoproteins. And in one of them, okay, so all this 130 milligrams is contained in lesser number of particles. All right, so this is just a simplified diagram. Whereas in the other, the same 130 is split among these multiple smaller particles. And clearly we know that if you have a large number of these smaller, denser particles, the so-called small, dense LDL, that makes it more heterogeneous. Okay? So ultimately, it's not the circulating cholesterol, but it is these LDL particles which go into the vascular endothelium and then start the atherosclerotic process. Okay? So if you have these large fat particles, these are happy. These are chubby particles. They just keep bouncing around, floating around. They don't cause much problem. But it's these small, dense, grumpy particles which actually burrow into the vascular endothelium and cause atherosclerosis. So you might have the same total amount of cholesterol, but if you have more of these particles, then we know that they are actually more heterogeneous. Okay? Now, none of this is just speculation, so we do have data on this. Okay? All right, this is another way of looking at it. Okay, so this thing between LDL-C and LDL-P, that is LDL cholesterol concentration, LDL particle number. Okay, so it's a little busy. So let's actually start with this. Just look at this portion. Okay, so if you take people, and this is what they have plotted is both the LDL cholesterol concentration and the LDL particle number. So you can actually count the number of particles. Okay, you can do NMR And then you can actually count the number of particles. So it's actually expressed in so many micromoles per liter and so on. Okay? So if you do this as a function of the triglycerides, all right? So the triglycerides. So as the triglyceride levels go on, then, so from 0 to 150, so that's like, I mean, when your triglycerides are normal, then as your cholesterol concentration goes up, your particle number also goes up. Okay? So they're actually similar. So somebody who's got a higher cholesterol has a higher number. But look what happens in people with higher triglycerides. When you have higher triglyceride number, particle numbers keep going up, but the calculated LDL cholesterol doesn't. So there is this discordance between the LDL-P particle number and LDL cholesterol concentration. So this is something that only happens in people with high triglycerides or in people with low HDL cholesterol. Okay, we know that that's usually it's high triglycerides which cause low HDL cholesterol. So the same thing. So if you have somebody with low HDL cholesterol, there is this discordance between LDL particle number and LDL cholesterol concentration. Okay? And not so in people with high HDL cholesterol or normal triglycerides. So in people with high triglycerides, you need to look at LDL particle numbers. Or another way of looking at it is, you can actually look at the ApoB numbers. Okay, so remember, there it is. So every one of these particles actually has a protein moiety, the ApoB. So instead of actually doing particle numbers, which actually is more sophisticated, it might be easier to measure ApoB. I think it's available in India, I think ApoB is here. So that's an easy test to get. So you can get ApoB, okay, which measures this. Okay? And as I was telling you, so this is data from the MESA study, okay, multi-ethnic study of atherothrombosis. So where they've actually looked at people who were concordant and who were discordant, okay, where there was concordance between the LDL-C and LDL-P versus those who were discordant. And if you look at those who are discordant, your LDL cholesterol concentration doesn't even predict cardiovascular risk. Look at the p-value. The only thing that predicts cardiovascular risk in these people is their LDL particle number. So again, there is some confusion about this. So should we be ordering an NMR spectroscopy in everybody? No, we don't need that. Okay, only in those where we expect there to be a discordance, and those are people with high triglycerides. So, like I see patients, specialist clinic in Mayo Clinic, so they come and they have FH. Okay, so in people with FH, there is no need to do anything else. I don't do coronary CT, I don't do this, that. LDL cholesterol is high, they are at high risk, treat them. It's a simple disease. Okay, it's these high triglycerides where it's a little bit. Now, so should we be doing this ApoB or anything in all of them? Actually, not even that. Okay, so there was this last thing that I did not highlight then, and that's the non-HDL cholesterol. Okay, so this is a poor man's ApoB and NMR spectroscopy, LDL particle number, all of these things. Okay, so in people with high triglycerides, just look at the non-HDL cholesterol. Okay, so that is a better marker of their atherogenic risk than LDL. And again, there is a lot of data on this. So this, I think, is the Framingham Heart study. So what you can see is that across any particular LDL cholesterol level, okay, all those with LDL cholesterol below 130, so you can see that increasing non-HDL cholesterol increases the risk for heart disease.